and welcome to my channel Biology by Katie. In this series of videos I'm making my way through the AQA A Level Biology specification. This video today is a little bit of a jump from the others because it is one of my most highly requested topics that I get in comments on social media. This is topic 5.1, photosynthesis. I really hope that it provides clarity to those of you that have requested it and to anybody else that watches it. Photosynthesis is a biological process that occurs exclusively within chloroplast-containing cells, meaning that it cannot occur in any cell that does not have a chloroplast. That is because chloroplasts contain the necessary products that are going to facilitate the light-dependent and the light-independent reaction, which we're going to take a look at today. Photosynthesis is such an important biochemical process in our lives because it is the process that converts light energy into glucose, and glucose we know is the primary respiratory substrate. First, taking a look at our chloroplasts themselves and the structure. So we can see here we have these series of flattened discs, fluid-filled discs on top of one another that are called the thylakoids, and we're going to be interested in the membranes of those thylakoids and the stacks of them. So the way that they're stacked on top of one another are referred to as the grana. We also have our liquid, our fluid that fills the chloroplast and surrounds the thylakoid membranes, and that is our stroma. So we are going to be looking at the process of photosynthesis in some detail, but it's worth remembering the overall formula, which is 6 carbon dioxide plus 6 water molecules, gives us 1 molecule of glucose and 6 molecules of oxygen gas. As I mentioned at the start there, the process of photosynthesis is split into 2 major reactions. The first one, the light-dependent reaction, and the second, the light-independent reaction, otherwise known as the Calvin cycle. As I'm sure you might be able to deduce, the light-dependent reaction is named so because it is directly dependent on the absorption of light energy to kickstart the reaction. On the other hand, the light-independent reaction doesn't require the direct absorbance of light, but it is dependent on the products of the light-dependent reaction, and so it is indirectly dependent on light. But we have split into those 2 reactions. Now, the light-dependent reaction occurs in the thylakoid membranes, so the membranes of those fluid-filled sacs that we saw when we looked at the structure of the chloroplast. And the light-independent reaction occurs in the stroma. Now, they do happen in a certain order, so the light-dependent reaction happens first, and then the light-independent reaction happens because, as I mentioned, the products from the light-dependent reaction are vital for the light-independent reaction. So the first thing that we're going to look at is the light-dependent reaction, the one that occurs first of all, the one that occurs in the thylakoid membranes. So it's important to look at what these membranes look like and what we have within the membrane that's going to facilitate the light-dependent reaction taking place. On the screen here, you can see a snippet of the thylakoid membranes, the functional unit of one of the thylakoid membranes. In this snippet, you'll be able to see a number of membrane-bound components that are going to be really, really important in the progression of the light-dependent reaction. First up on the diagram, we have got photosystems, so the green proteins that are spanning through the membrane. We've got photosystem 1 and photosystem 2. Now, what photosystems are is they are complex groups of proteins and chlorophyll, so light-absorbing pigments. And like I said, there are 2 of those in the membrane. Photosystem 2 is the first one that we come across, and then photosystem 1, the second one, a little bit further along the membrane. We also then have a series of membrane proteins that are involved in the transport of electrons. So they are called the electron transport chain. What they're responsible for is picking up free electrons and transporting them through the membrane to their final destination. In the centre, in blue, we have the hydrogen ion, otherwise known as proton pump. This is a carrier protein that's responsible for the active transport of hydrogen ions against a concentration gradient. And then finally on the end, we have enzyme ATP synthase that's responsible for catalyzing the phosphorylation of ADP into ATP. So let's start with the very first step of the light-dependent reaction, which is the photoionization of chlorophyll. Now, we've already identified chlorophyll as being the photosynthetic pigment within the photosystems of the thylakoid membranes. So they're responsible for the absorption of light energy. The term photoionization itself, if we split it up, so photo refers to anything to do with light, and ionization refers to when a molecule or an atom becomes ionized or charged, meaning that usually it either loses or gains an electron to become either positively or negatively charged, respectively. In this case, the chlorophyll loses an electron and becomes positively charged, so let's have a look at how that happens. First of all, some light energy is absorbed by the chlorophyll molecule, or a chlorophyll molecule, within photosystem number 2, so the first one that we come into contact with in the thylakoid membrane. As this happens, the electrons within the chlorophyll molecule move to a higher energy state, and one of them is able to break free from the chlorophyll altogether. When a substance or molecule loses an electron, that substance or molecule is said to have been oxidized and to be positively charged. So the chlorophyll molecule is now positively charged and that electron has been released. That electron is then going to be picked up by the first protein in the electron transport chain. As the name suggests, the electron transport chain is a series of proteins that act as a chain through the membrane. So when an electron is picked up by the first protein, it is then passed along almost like a conveyor belt to the next one and then the next one in the series. Each of these proteins in the electron transport chain is said to be of lower energy than the previous one. So as the electron is travelling along from one to the next, it loses or releases a small amount of energy. This energy will be used by the proton pump over which it passes to actively transport hydrogen ions against their concentration gradient. The fate of that first electron is that it ends up being taken up by the second photosystem in the membrane, which is photosystem 1. The next step is a process that is unique to photosynthesis, and that is the photolysis of water. Now, we've seen the term lysis, or lysis as it sounds, as a ending of a word before. So hydrolysis, meaning breaking of bonds using the addition of water, and it has the same meaning here. So again, photo meaning light and lysis meaning to split. So what photolysis means is the breaking of bonds using light energy. The process of photolysis is an absolute essential requirement here because, as we have just discussed, an electron has been lost from a chlorophyll molecule within photosystem number 2. So as that has become ionized or positively charged, its function has been altered. It's not acting in the same way that it usually is or that it needs to be doing to continue with the light-dependent reaction. So ultimately, the electron needs to be replaced. The electron that was lost needs to be replaced. Light energy is used to split water molecules, so H2O, into 2 atoms of hydrogen and 1 atom of oxygen, and the hydrogen atoms are then split further into an electron and a proton each, or an electron and a hydrogen ion. So a hydrogen atom is a very, very small atom, the smallest atom in existence, made up of only 1 electron and 1 proton. So once it becomes ionized, we say that it is made up of just 1 proton. So that's why we refer to it as a proton. And we also have the release of electrons as well. 1 water molecule, therefore, will yield half an oxygen gas molecule, 2 electrons and 2 protons, or 2 hydrogen ions. The electron will then be used, or one of the electrons will then be used to neutralise the chlorophyll molecule that has been positively charged, that has been ionised, and the hydrogen ions have another role within the light-dependent reaction. There are 2 equations for photolysis that are accepted by the specification. You can either split up 1 single molecule of water and end up with half an oxygen gas molecule, or you can double up so that you end up with 1 molecule of oxygen towards the end. The next step of the light-dependent reaction is the active transport of hydrogen ions against their concentration gradient into the intermembrane space, the thylakoid space. So as we mentioned before, the proton pump that is embedded in the thylakoid membrane is powered by the energy that is lost as the electron that was released from photosystem number 2 passes along the electron transport chain. We know that energy is required by active transport to move molecules or atoms against their concentration gradient. So that's what's going to happen here. We have an accumulation of hydrogen ions in the stroma, and they are immediately actively transported into the thylakoid lumen. The reason for this is we are trying to generate a proton gradient or a hydrogen ion gradient inside the thylakoid lumen, where there is a high concentration of hydrogen ions in the lumen in comparison to outside in the stroma. The reason for this is that generating a high concentration of hydrogen ions inside the thylakoid space means that naturally the hydrogen ions or the protons will want to diffuse back into the stroma through a membrane-bound protein. And they can. That is where we have our ATP synthase coming in. So ATP synthase is a membrane-bound enzyme that also acts as a hydrogen ion channel protein. It allows hydrogen ions to diffuse via facilitated diffusion from the thylakoid lumen, where we have generated a high concentration gradient, into the stroma, so down a concentration gradient. And as each hydrogen ion passes through the ATP synthase channel, the energy from that is used to drive the phosphorylation of an ADP molecule into an ATP molecule. And therefore, we have the synthesis of ATP, which is the first product of our light-dependent reaction that is going to be used in the light-independent reaction. As we mentioned at the start, there are two photosystems embedded within the thylakoid membranes, meaning that there is a second opportunity for light to be absorbed. A very similar process happens here at photosystem number 1, photoionization. So a photon of light energy is absorbed, an electron enters a higher energy state and breaks free from the chlorophyll molecule, meaning that the chlorophyll has become ionized or positively charged. Now, due to the continuity of the light-dependent reaction, by the time the photosystem number 1 is ionized, or the chlorophyll within photosystem number 1 is ionized, the electron that has been released from photosystem number 2 and has been carried down that electron transport chain arrives at photosystem number 1 to neutralize it. The electron that has just been released from photosystem number 1 is picked up by a final protein in the electron transport chain before it is passed on to a nearby electron carrier called NADP. As NADP gains an electron as the result of this, we refer to it now as reduced NADP. It's been reduced by the electron that has been passed to it from the electron transport chain. Just a little bit of a recap then about the products of the light-dependent reaction. So we've got oxygen, our first product that was produced during the photolysis of water. Now, oxygen is considered to be a waste product of photosynthesis, so it will diffuse out of the stomata and be released by the plant cell. Then we have got our ATP produced by chemiosmosis, the movement of hydrogen ions against their concentration gradient. And finally, we have reduced NADP, so where NADP was past that final electron from the electron transport chain. ATP and reduced NADP will both move into the stroma, where they will be utilised in the light-independent reaction. The next part of photosynthesis that follows on from the light-dependent reaction is, as we mentioned at the start, the light-independent reaction, which occurs in the stroma, which is the liquid that surrounds the thylakoids inside the chloroplast. As I mentioned at the start, the light-independent reaction does not involve the direct absorption of light, but it does rely on the products of the light-dependent reaction. So the ATP and the reduced NADP that were produced in the light-dependent reaction are going to be used in this next stage. Another important substance that the light-independent reaction does need to progress is carbon dioxide gas. Carbon dioxide enters the plant through the microscopic pores in the leaf called the stomata, and it will diffuse through the tissue layers of the leaf until it reaches the chloroplast. The light-independent reaction is also sometimes referred to as the Calvin cycle. This is an acknowledgement of the scientists who originally identified this series of reactions. And of course, the word cycle tells us that this particular reaction is cyclic, and we will start off with the same product that we end up with. So the original product is regenerated by the end of the reaction. So the first molecule that we're going to look at here is ribulose biphosphate, commonly known as RUBP. It is okay to call it RUBP in exam style questions, you will get the mark. This is a 5-carbon molecule, and that's what we need to know about that. It has 5 carbons. We don't need to know the rest of the structure. The first step of the Calvin cycle is a process that we refer to as carbon fixation. And what that means is the carbon atom from carbon dioxide gas that has diffused into the chloroplast is incorporated into a new molecule. So it's added on to the ribulose biphosphate, the RUBP. So this 5-carbon molecule becomes a 6-carbon molecule. And this process of carbon fixation is catalyzed by a specific enzyme called rubisco. And rubisco will be found in the stroma of the chloroplast, ready to catalyze this particular reaction. Now, the 6-carbon molecule that has been produced as a result of carbon fixation is a very, very unstable molecule, and therefore it exists only momentarily before it splits into 2 3-carbon molecules, glycerate 3-phosphate, commonly known again as GP. Again, that's absolutely fine to put in the exam, GP. The 2 molecules of GP are then reduced into 2 3-carbon molecules, still 3-carbon molecules, but they are now called triose phosphate. They've been changed into a new substance called triose phosphate, otherwise known as TP. So the reduction of glycerate 3-phosphate into triose phosphate involves the products of the light-dependent reaction that were produced in the thylakoid membranes. The reduced NADP that was produced in the light-dependent reaction provides the reducing power, so the necessary substance to reduce GP into TP, and ATP provides energy when it is hydrolyzed to complete this conversion. So it's just worth mentioning here that if the light-dependent reaction hasn't taken place in the absence of light, for example, then we wouldn't have the necessary products to reduce GP into TP, and the Calvin cycle would not be able to continue. So we have 6 carbons available. It's worth remembering that this is a cycle, so we must retain 5 of those carbons from the 2 triose phosphate molecules in order to regenerate RUBP. So for each turn of the cycle, we can only afford to extract 1 carbon to produce organic molecules, organic substances such as glucose. So one of those carbon atoms from all 6 of the 2 triose phosphate molecules is removed at this point from the cycle and is used to build large organic molecules such as glucose, whilst the remaining 5 carbon atoms will return to the start of the cycle. They will be used in the regeneration of RUBP, which of course was our original 5-carbon molecule. The regeneration of RUBP from the carbons belonging to triose phosphate involves energy from the hydrolysis of ATP. So again, this is another instance where ATP produced in the light-dependent reaction is used in the light-independent reaction. Let's just recap then at that entire series of reactions as a complete cycle. We start off with our RUBP, ribulose biphosphate, which will be found in the stroma of the chloroplasts. Carbon dioxide that has diffused into the chloroplast will be fixed, so the carbon from carbon dioxide will be fixed and added to the ribulose biphosphate, converting it from a 5-carbon molecule to a 6-carbon molecule. This reaction is catalyzed by enzyme rubisco. This 6-carbon molecule produced is extremely unstable and splits immediately into 2 3-carbon molecules of GP. GP is then reduced into 2 molecules of TP, and this process requires the reducing agent from reduced NADP and energy from the hydrolysis of ATP. Remembering that both of these substances came from the light-dependent reaction originally. Then we have the idea that 5 of these carbons belonging to the 2 triose phosphate molecules must be used in the regeneration of ribulose biphosphate, RUBP. This process of regeneration requires energy from the hydrolysis of ATP. The final single remaining carbon is removed from the cycle and is used to build large organic compounds such as glucose. The fact that we can only afford to extract 1 carbon atom per cycle means that there must be 6 turns of this Calvin cycle in order to produce 1 molecule of glucose, as glucose is made up of 6 carbons in total. For the final part of this topic, 5.1 photosynthesis, we've got to identify any external factors that may have an effect on the rate of photosynthesis, so how quickly these two reactions occur. The first environmental condition that can potentially have an effect on the rate of photosynthesis is light intensity, the amount of light available to the photosynthetic cell. We know specifically that the light-dependent reaction involves the absorption of light by chlorophyll molecules, which kicks off the series of reactions that follow. If you look at this graph, there does seem to be initially a positive correlation between light intensity and rate of photosynthesis, suggesting that the higher the light intensity, the faster the rate of photosynthesis gets, until we reach a certain point where the curve seems to flatten. At this point, we would say that there is something else limiting the rate of reaction, limiting the rate of photosynthesis. It is no longer light intensity or the amount of light available that is slowing down the maximum rate of photosynthesis. It's worth remembering that any light absorbed by the plant must be the correct wavelength. So they can only absorb wavelengths of lights that are in the red and blue spectrums of the visible light wavelength spectrum. They cannot absorb green light because it is reflected by the chloroplasts. The second potentially limiting factor on the rate of photosynthesis is the carbon dioxide concentration. Carbon dioxide is used to convert RuBP into the 6-carbon molecule that ultimately starts the turn of the Calvin cycle. So the more carbon dioxide that is available to the plant, that is in the stroma, the faster the Calvin cycle will be able to progress. Much like light intensity, there is initially a positive correlation between carbon dioxide concentration and rate of photosynthesis because of this, but again, there is a point at which the reaction flattens, the curve flattens, because something else becomes a limiting factor. It is no longer carbon dioxide that is preventing the rate of photosynthesis from reaching its maximum. The final condition that we're going to look at that has an impact on the rate of photosynthesis is, of course, temperature. Now, the graph depicting the relationship between temperature and rate of photosynthesis does look slightly different to the other two. At first, again, we can see a positive correlation between the temperature and the rate of photosynthesis. During this time, as temperature increases towards the optimum, the molecules inside the plant cell will be increasing in kinetic energy, and therefore they will be colliding with one another more frequently, meaning that we are likely to have more successful enzyme substrate complexes being formed, resulting in an increase in the rate of photosynthesis. However, if the temperature continues to increase far beyond the optimum, the enzymes will begin to denature, meaning that they are no longer functional and cannot catalyze the reactions involved in photosynthesis, meaning that the rate of reaction will ultimately drop and eventually come to a complete halt. So for the last slide, let's take a look at what the AQA specification says that we must know for this topic, 5.1 photosynthesis. First of all, we have to know the steps of the light-dependent reaction to include the photoionization of chlorophyll, where an electron breaks free from a chlorophyll molecule due to the light energy absorbed. The production of ATP via the chemiosmotic theory, the chemiosmosis process, where hydrogen ions are actively transported against their concentration gradient and then pass back through the ATP synthase, and also the reduction of NADP, so where we have that final electron being passed to NADP, causing it to be reduced. The mechanism of the electron transfer chain itself, so the movement of the electrons through the membrane down this network of proteins. And the photolysis of water, where we broke water molecules down using light energy to release hydrogen ions and electrons. We then must be aware of the processes within the light-independent reaction, starting off with the reaction between carbon dioxide and RuBP to ultimately produce 2 3-carbon molecules of GP, and the fact that this reaction is catalyzed by enzyme Ribisco. We then have the reduction of GP into TP using the products of the light-dependent reaction, namely ATP and reduced NADP. The fact that the majority of the carbons from the 2-triose phosphate molecules are used to regenerate RuBP using energy from ATP, and that some of them, or one of them, is used in the production of useful organic substances such as glucose. So there we have it, topic 5.1 complete. I hope this has been a clear and useful video for all of you and that it's helped you with your understanding of photosynthesis. Thank you.